The last presentation before lunch is to give an update on the Black Swan project. We get a lot of questions about that, and it's linked in with this ASCENT trial that we've touched on a couple of times, which is the trial to try to have the best outcomes for patients with high-risk smoldering myeloma. And so the fundamental part about the Black Swan project is to give the best therapy that we have to patients at that earliest time point to achieve the best results and hopefully start to have very long remissions and hopefully to start curing myeloma. And so this is a project that's been going on now since 2012, so it's been going on for a few years. And so what I'd like to do is to give a little bit of an update. And so the main initial part of this project was to introduce tests for minimal residual disease. And so if you're trying to achieve a fantastic outcome and have a deep response, you need to have methods to measure myeloma at a low level in the bone marrow. And so the first part of this project was to develop and introduce and start using methods for minimal residual disease testing. And so there are two ways to do this. One is using a genetic method that's called next generation sequencing. And another method is to run the bone marrow sample through a flow cytometer where you have the cells flow through the machine and the machine can detect the myeloma. And this is called next generation flow, NGF. So you have two methods for MRD detection that were developed in parallel with this Black Swan project, and those are what we use to assess how the patients are doing with early treatment. Let me go forward from there. So the first question that comes up is this the goal for every patient? Are we immediately trying to achieve cure for every patient? And so the answer to that is absolutely no. There was an answer to a question from a gentleman back here who said that his protein was steady at 0.39, and he asked Dr. Abanar whether he should take more treatment to do better, and we said no. Right now, we are attempting to achieve cure for some patients, particularly patients with high-risk smoldering, but we're not recommending that for everyone until we see how well the patients are doing in these early trials. And so, yes, it's a goal, but it's not what we're recommending for everyone right now. And then, in response to a question yesterday, someone asked, well, should they be getting MRD testing? And I think that maybe you could get it for a couple of reasons, but it's certainly not mandatory. So there are 4 reasons that you might think about getting MRD testing, and these testings can be sent to different labs. There's an Activism company that runs this NGS method, and then the flow method is also available at different labs. So both the flow and the sequencing methods can be, so your doctor can send the samples to get this testing done. The first reason to do it would be to see if it's negative, it would mean that you've had an excellent response and your outcome could be somewhat better than average. And so this is what's called a prognostic test, where it's indicating that you're doing a little bit better than average. A second one, which we are mostly interested in, is to use it as an endpoint within clinical trials where we're giving treatments where in the trial we're trying to achieve MRD negative. So the purpose of the trial is to get to that MRD negative, particularly at that 1 in a million level, which is this 10 to the minus 6. So what that means is that you measure, you test 1 million cells, and none of them are myeloma cells. And so that's called negative at 10 to the minus 6. So we're aiming for that. And so what we're looking at is if that is happening with a particular treatment and not with another treatment. So if you have 2 treatments and one is giving you MRD negative and the other one is not, then this could be a better endpoint. And so right now we are working with the FDA to submit trial results, and we are hopeful that the FDA will accept this as an endpoint, so that with new treatments coming along, for example, CAR-T cells, if CAR-T cells produce MRD negative at this 1 in a million, 10 to the minus 6 level, that could be better than some other treatment which is not achieving that. But that's a use within a trial. The third thing is for decisions. And so we need more information to really send off the test, and then you get the result back, and then you actually make treatment decisions. So we mostly don't know enough to make a secure decision. There are a couple of examples that have been mentioned. So for example, if you've taken VRD and you are MRD negative, what we're hearing is that for patients who've had a fantastic response, it may be that you don't immediately need to have a stem cell transplant. And so that's a possible decision. Another possible decision is if you are taking Revlimid maintenance and you become MRD negative, maybe you could stop the maintenance. However, we really don't know that, because the opposite way of looking at that is if you're going along, you take your Revlimid maintenance, and you become MRD negative, that actually means the Revlimid's working. And so do you want to stop it because it's working? Or, we don't know. Maybe if it's working, that's the exact situation where you should stay on it. And so we need studies to use it in that way. And then the fourth one, which is part of the focus of the Black Swan project, is that we're using it as a test to guide us to see if we're moving towards achieving a cure. So the reality check is, how close are we to having this kind of very successful therapy? And so I mentioned already this morning that in this SWOG trial, over half of the patients are doing well at 7 years. So just with VRD, we're moving to longer survival. And then if we look at that in a little bit more detail, we can see who are the patients that are living longer just with this 3 drug, the Velcade, Revlimid, and Dex. Who are those patients that are doing the best? And so the patients who have had at least a VGPR, now you'll notice here I'm not saying MRD negative, I'm saying if they've had just like a VGPR, so you could be a patient who has a protein level of 0.39 or a very low level. You could have a VGPR. 60% of the patients who have a VGPR or better are doing well at 8 years. So what we're saying is that patients with deep response, even short of MRD negative, are starting to have these longer survivals in the 5, 10 years and beyond. Now, we also have heard that if you have VRD plus the transplant, you can get even a deeper response, and you can get those responses at the 10 to the minus 6 level, and those are the patients that will stay in remission even longer. And so the reality check is that just with VRD and transplant, we are starting to see these much longer remissions. And so the next question is, if we enhance that treatment a little bit and give it to patients a little bit earlier at the high-risk smoldering stage, how much better can the outcomes be, and maybe will we start to be able to cure some patients? And so this is just to show you that we are working with the FDA as we move forward and trying to get approval for the use of MRD testing as an endpoint, and we're gathering together trials, all these different trials in the green box there, and we did have a meeting with the FDA in December, and it looks like MRD is on track to be something that the FDA will accept for monitoring in the trials in the way that we are proposing. So we're optimistic about that. The flip side of this is that if you are doing a great job, what are the patients like, the ones that are not responding well? And so the Black Swan project is to try to do the best job for the patients who can be treated right now, but also to understand why patients may be not doing so well. And so in the Spanish study where they used VRD and transplant, out of the 225 patients who had a fantastic response, there were 6 relapses. And so the question is, what is the problem for patients who are failing with our current best therapies? If someone's failing with BRD and a transplant, what's the story with their myeloma? And what you can see here of those 6 patients, there was this predominance of this 1Q plus. And this is one of the reasons that we've started to pay more attention to that, because that does seem to be something that's showing up in patients who are relapsing after having gone through our best therapies. And we also know that if you look at those, if you follow those patients along for a while, the patients that have that 1Q plus, they have what is called here subclonal mutations, is that they start to mutate more. And so the idea is that you don't want to let them sit around for too long because they could be messing around in there and doing bad things. So basically, when you see these, we have the idea that it's important to probably jump in and do something sooner rather than later in those patients who are either failing or relapsing early. And so if you look visually about what we are trying to achieve with the Black Swan project and with the CURE trials, we have 2 trials right now, which I'll explain in detail in just a second. We have the CESAR trial and the ASCENT trial. And basically with those, we are able to get rid of most of the iceberg. This is the picture that Jean-Luc showed you, where the myeloma is above the surface, but a lot of it is at the bottom of the iceberg. And our problem is, how far do we get down the iceberg with our treatment? And you can see here that we think at that 10 to the minus 6 cutoff, that red line, it's probably taking us most of the way down the iceberg, but we're not really sure. We know for sure that just with the SPEP, we're not getting to the bottom of the iceberg, and we're not really sure if we get down to 10 to the minus 7, 1 in 10 million, maybe we would be down to the bottom of the iceberg. But we know we're doing a pretty good job. And so with the CESAR trial, this is a trial that started in Europe, and this is a trial for smoldering myeloma, high-risk smoldering, and the treatment here is KRD, and this is a treatment that Jean-Luc mentioned, Kyprolis, Revlimid, and Dex, which seems to be a little bit better than Velcade, Revlimid, Dex. And this is the protocol where he said that if you take 12 cycles of that, that actually could be really good, and maybe you don't even need a transplant. But in this study, there were 6 cycles of KRD, the 3-drug regimen, and then a transplant, and then a consolidation and maintenance. And as Dr. Abenour mentioned, this treatment is finished after about 2.5 years. And so the idea is to give about 2.5 years treatment and then stop and see how good is the benefit that you've achieved. And so what do we have so far? Well, the treatment has been extremely well tolerated, and Dr. Mary V. Mateos from Salamanca, she's presented her results a couple of times, and the vast majority of the patients, 94% of the patients, are now in remission out at 3 years. And so those patients have been achieving MRD negative and at the 10 to the 6th level, and they're moving out forward without any pattern of early relapses. And so it's really pretty encouraging. And so the US trial, which is the ASCENT trial, started about 6 months ago, and in fact, Dr. Abenour put the first patient into that study, and that patient has done very well with a very deep response. The treatment in this trial is daratumumab plus the KRD, so this is a 4-drug regimen, and the trial has started with that as the therapy, with the option moving forward that some patients may receive the autologous stem cell transplant. You see there a dotted arrow. And this is related to the guidance from the FDA, where and this question was actually asked to me by the lady over here on the left, how has this treatment been tolerated? So this is a 4-drug regimen. That's a pretty strong regimen. Are the patients able to handle that well? And I'm sure Rafat could comment on this, but mostly the patients have been handling this in a pretty good fashion, and there hasn't been any untoward problem in the study thus far. But again, the idea here is to complete a total of 2 years therapy and to assess how many patients will achieve that MRD negative, and we're expecting at least 60–70% of the patients will get down to that 10–6 level, or maybe better even. So we're pretty optimistic about that. And so with the CESAR trial, right now it's running at 62% of the patients are MRD negative, and we think that the addition of the daratumumab will push us up to at least 75%. And so we are really just waiting to see how that will go, and this will be the treatment for patients who have sensitive disease who are going to be responsive to that regimen. However, along the way, we're studying the patients who are not sensitive and may have resistant disease even with that therapy, and we know quite a few things about those patients, particularly about the different antigens that are on the surface. And the myeloma can have these different things like number 38, which is what daratumumab is directed against. So daratumumab is anti-CD38. And then I mentioned here CD46. That's one which is in patients who have that 1Q+, and there's a new monoclonal antibody working against that. So there is an idea to introduce therapies for the patients who might be resistant. And we already know, and it's been up in discussions, that more broadly we could be thinking about something like a CAR-T therapy that could be broadly active in those resistant type patients to mop up the residual cells in patients who are failing or relapsing or have persistent disease in the frontline therapies. 88% in this particular study that was presented at ASH in December, 88% of the patients responded with the CAR-T cells against that BCMA, which is on the surface of the myeloma. And that those patients who achieve MRD negative have actually been having sustained remissions. Now, we know, unfortunately, and the lady over here got the BB2121, some patients do have early relapse, but the patients who have the MRD negative, in general, are staying in remission a whole lot longer. And so the broad approach that we have within the Black Swan is to start early and give the best therapies that we have. For example, the CESAR trial, the ASCENT trial. We also have a similar trial that's going on in Iceland, which we're going to talk about just in a minute here. And then using different protocols in an effort to eliminate residual disease or new disease in patients coming out of those studies. And so the motivation for that is based upon new data where even in the relapse setting, and I think that this is tremendous news for everyone, is that we have been using these daratumumab combinations. For example, daratumumab Velcade-dex or daratumumab Revlimid-dex, and what has been shown in a couple of trials, which are called the Castor and Pollux, the twin trials, that patients who get to have an MRD negative, even in this resistant or relapse setting, can do very, very well. So we're optimistic that even trying to focus on these resistant or relapsing patients, we can move towards having excellent outcomes. Now, a key part of the project, and this is obviously something that patients are pretty involved with, is the testing of the MRD. And so the first question is, if you're MRD negative, how are you going to monitor that? And then that immediately sounds like a lot of bone marrows, right? If that's in the bone marrow and then we're going to monitor it, that means there's going to be more bone marrows, right? And so we have been quite interested in coming up with testing that would allow us to assess what is going on using the blood. And so studies at the University of Salamanca have been showing us that if you monitor in the blood, even looking at patients who are in complete remission, in this particular slide here, you can see as you move from that CR, that yellow box there, if you look down, the patient who was in CR here had CPC. So there were some red dots that were floating around in the blood at that time. And so even although that patient was in a complete remission, when we tested that patient's blood, we found myeloma in the blood. Now this patient did fine for 14 months. But after 14 months, you can see those CPC red dots were building up. And as you track over to the right, you can see after 14 months, there was a lot of those red dots over there, and the patient had clearly progressed and relapsed. And so now we know that if we find these cells in the blood, then that can be an early indicator of early relapse. And so the Spanish have found that there are about 15 to 20% of patients who are in apparent complete remission or stringent complete remission who have those cells in the blood. When they did their initial testing, it was 7 out of 47 patients. And then over on the right, you can see that those are the patients that relapsed or progressed within an average of 9 to 10 months. And so we've learned that there are different MRD patterns when you look at the bone marrow and then the blood. So the ideal situation, which we're trying to achieve in the high-risk smoldering patients, is negative in the bone marrow and negative in the blood. But what we've discovered is that there are some patients who are positive in the bone marrow, but then they're negative in the blood. And those are patients who seem to be able to stay like that for a long time. Those are not patients who would relapse right away. However, there are patients who are positive in the bone marrow, and then there are those patients who are also having cells in the blood, and those are the patients who have early relapse, and the ones that we need to be targeting early to think about introducing treatment. And so the idea here is just like treating early disease, high-risk smoldering early, in the relapse setting, we would also focus on trying to treat those folks early and achieve the best results in that relapse setting. And so the new concept is to retreat what would be called biochemical relapse. And so this would be based upon the early findings of myeloma cells in the blood, but also using this new sensitive mass spectrometry method to pick up low levels of myeloma protein. And so to introduce treatment early in a trial setting, and also to study what the mutations are of those cells which are in the blood and the bone marrow as a basis to select the therapy. And so broadly, what we're thinking is that with the CESAR and the ASCENT trials, in the sensitive cases, we will be able to get rid of the iceberg, we hope. However, we realize that in at least 25% of the patients, and maybe more, there will be cells that are remaining, and we're going to need new therapies to introduce to get rid of the rest of the iceberg. But we are hoping that the iceberg will be gone with some of our newer ideas. And so we're moving towards this future model that I've presented now earlier, where we will be having therapy which is for myeloma that would be used for the smoldering cases, and then also for biochemical relapse later in the disease, where we're trying to achieve the best results by introducing the therapy as early as possible. And then setting aside patients where we're comfortable to just observe those patients. Hopefully that gives you a good flavor of what we're trying to achieve. I think that particularly with the CESAR and the ASCENT trials, we're making good progress to show what is the value of this approach. And certainly we've been able to set up the tools that we need for the early monitoring and the ongoing monitoring so that we can understand how the patients are doing and what is being achieved. But we are particularly excited about one aspect of the Black Swan project, which is going on in Iceland, where we're monitoring the early disease as a basis for understanding the molecular mechanisms and the immune mechanisms that are related to the evolution of the disease and then the transformation of the disease into the active stages. And so maybe we can go ahead and show the short video about Iceland which we just made. It's a new one which we made based on our visit there in November, just at the end of the year. We can just play that, please. Yay!
SMM study is Iceland screens, treats, or prevents multiple myeloma. And the whole idea is early detection can lead to early treatment and therefore prolonged survival, improved quality of life, and hopefully cure. We invited all Icelanders 40 years and older to participate in the study. This is the third year, and we have very good participation rate of over 80,000 people. And now we have almost 50,000 samples in-house that we have analyzed. Those that are diagnosed with MGUS or smoldering will enter a randomized clinical trial with 3 different arms with different approaches with regards to workup and follow-up strategy.
Without a doubt, this is the largest and most ambitious study in the area of myeloma and plasma cell disorders. And the interest for the Black Swan Research Initiative is that early diagnosis can lead to the possibility of early treatment, early intervention, and to potentially be able to cure myeloma. Because it is nationwide, the conclusions that you can draw are much more decisive and much more important. What are the features that would indicate the progression from MGUS to smoldering to myeloma? For a whole nation, it's just incredible.
I'm GM Durie.
Nice to meet you.
Pleasure to meet you.
People here in Iceland understood that it was our duty to help science in this way. Your organization is a testimony to the fact that we can and should and must use all means to advance in the fields of science. Just like me.
People of Iceland really show a willingness to help others to go into these trials and research programs because they know that it's going to open up the door to the cure. And that's our ultimate goal always, is to find a way to a cure.
I'm very proud of it, really. And for us here, I think it's a natural thing to, at least from my perspective, is to participate in such research.
It's very important for us all, so I think it's our duty to do it. So I feel very happy about that.
Despite all these screening programs in the world, there is very little research on the psychological impact of being screened. From the very beginning, this has not just been a very narrow research-minded project, but it has always been built on the idea that this is being done for the patients. And I think that's one of the reasons why so many Icelanders signed on to the study, that they realized that this was really being done to take really major steps towards treating myeloma.
Since we opened the clinical site last year in April, we have met 1,200 participants for the first visit.
And we do blood tests from everyone, and for most of them we take some bone marrow tests as well, and then we do some physical examination.
For the screening to be useful, it needs to be practical and manageable, and so you do need a bone marrow for the initial diagnosis, but subsequent to that, it's now feasible to use blood testing to monitor what is the status of the MGUS. Is it remaining stable? What is the status of the smoldering myeloma? Even the myeloma with treatment can be monitored and followed using blood testing.
The prevalence of smoldering myeloma is higher than we anticipated. So that could have some implications throughout the world, because treating early at the smoldering phase is beneficial. You don't find people with smoldering myeloma because they are asymptomatic unless you screen for them or if you find them by chance. And we don't believe in a chance here. We want to go out there and find them and offer them early treatment.
The 80,000 and more individuals who are participating have genetic information available. They have been sequenced, and this genetic information can tell us if there are underlying genetic variants in patients with plasma cell disorders that are not present in the rest of the Icelandic population. And it will be possible to develop a risk model of genetic features that predispose people to get MGUS and myeloma.
As a patient, this research is critical for patients going down the road further. Just sitting through the meetings here in the latest on the iStop program is very exciting, and the opportunity for us to bring that information back to support groups is unbelievable.
When you gather so much information, blood samples, results from the hospitals, all cancers, every prescription that they have gotten, you see that the complexity of the study becomes more and more. But also you see so many opportunities on what can be done. So every time we sit down and talk about new ideas on what to do, we come up with 10 or 20 new ideas. So what we're doing now is, besides what we decided at onset, we're also looking at other important areas like comorbidity, like neuropathy, like cardiac disease, renal disease, and so on. So the opportunities are actually endless.
So I hope you enjoy that. We're obviously very proud of our ability to collaborate with Sigurdur Christensen, who is obviously a wonderful guy, the head of the blood disorders group at the University of Iceland. And he now has close to 20 people working in this team focused on MGUS, smoldering, and myeloma, doing all of the basic lab work, the molecular work, the immune testing work, and then, as he said, now moving into the treatment phase for the patients in Iceland. So I don't know if any immediate comments or questions which lie between you and lunch.
Very quickly, the Icelandic population is homogeneous. And if so, which I assume it is,
yes.
How can you? Will it be easily extrapolatable to other less homogeneous societies?
Right. So this is a key question, and so the two sides to this are which we've actually been studying around the world, which is that basically myeloma is myeloma. In other words, the genetic changes, the FISH testing and all of that kind of stuff within myeloma is, that does seem to be relatively standardized around the world. What is different is the predisposition. And so, for example, we know that African Americans are 2 times as likely to get myeloma. And that means that there are certain aspects of the immune system or something that is allowing African Americans to get myeloma. And so the idea is that we can definitely study the myeloma, and that's going to be consistent around the world, but there will be an opportunity to compare the Icelandic population with the African American population, with the Asian population, which we have an Asian myeloma network, a clinical trials group in Asia. And so actually we will have the opportunity to understand the differences between different populations that might lead to that different susceptibility factor. Yeah.